Okay, today we're going to be talking about DNA and RNA. As you can see here, there's a couple different diagrams for DNA. On the far left is a kind of a, what we call maybe a simple picture of DNA, but you can see that it looks like a double helix or a twisted ladder. Then you have a more molecular picture in the middle. The double helix looking diagram shows you that in the middle you have something called base pairs. Your top 4 clients are the base pairs. The base pairs are adenine, thymine, guanine, and cytosine. Adenine always pairs with thymine, so you should see in that diagram that the yellow is always with the green. Guanine always pairs with cytosine, but the red always is with the orange. The outside part is known as the backbone to DNA, which is kind of bluish in color in the left-hand diagram. That is always the same. The backbone to DNA is always made up of an acid and a sugar, the sugar being deoxyribose. So you can see this as a double helix or twisted ladder is how some people like to describe it, or a spiral staircase. In the middle picture, you have A is blue, T is red, C is green, G is purple, A is adenine, T is thymine, C is cytosine, G is guanine. Those again are the base pairs. The base pairs are always found arranged in the middle of DNA. The backbone are on the sides, and the backbone in the middle diagram, the circle would be the acid, and the 5-sided diagram there is the sugar, because it is a 5-carbon sugar known as deoxyribose. Deoxyribose is the sugar found in DNA, and that's what helps form its name, deoxyribonucleic acid. DNA is made up of many nucleotides hooked together, so that's kind of where that nucleic comes in, and the acid comes from that acid piece. And again, in the diagram, the circle is the acid, the 5-sided diagram is the sugar, deoxyribose, and in the middle you have your base pairs. And the base pairs are in a certain order, and that order is very important because that order helps represent a gene. And the genes come together to make you who you are. So the order of the letters is a code, and it's a code for the gene, which are found on your chromosomes. Okay? DNA, like I said, is made up of nucleotides. Here is one nucleotide, another nucleotide, third nucleotide, and so forth. So each side is made up of nucleotides. A nucleotide is made up of an acid, the sugar, and the base. Acid, sugar, base. Acid, sugar, base, and so forth. And the 2 strands are held together by weak hydrogen bonds. Like I had mentioned, the important part are the letters and the order of the letters. So when we write out DNA, we're not going to worry so much about the backbone because the backbone is the same all the way down DNA. What is different are those letters or those nitrogen bases. So how you will write out DNA would be something like this. And I draw that little dash to represent the weak hydrogen bond. And hopefully you could write in the right-hand side to that DNA. You should know that A always pairs with T, G always pairs with C, and C always pairs with G. So there would be one segment of a DNA, or one segment of your gene. Okay. If I haven't mentioned it already, DNA is 2-sided or 2-stranded. Okay, DNA arrangement. When I say look at the handout, I kind of mean that page right before this. So I mentioned the nucleotide made up of a phosphate group. I called it the acid. The sugar and the nitrogen base. The phosphate group and sugar are arranged on the outside of the double helix, known as the backbone. That's the part that stays the same. The nitrogen bases are arranged in the center of the double helix. The guanine, the adenine, the cytosine, and the thymine, G, A, C, and T. Those are those bases, and the code. They represent the code. They represent your genes. The order is very important. I already mentioned that adenine always pairs with thymine, and cytosine is always with guanine. And so the A-T together is known as a base pair, and the C and G together are known as a base pair. So if I ever ask what are the base pairs and you just write out A, T, C, and G, that would be wrong. It's a pair. It has two. So A and T are a pair. C and G are what pair together. And I already mentioned that there's weak hydrogen bonds holding the adenine to the thymine and to the cytosine to the guanine. And you may think there should be a strong bond holding them together, but there's not. It's a weak bond. Because if you remember, when your cell gets too big, it needs to divide. And if it's making two new cells, each cell needs a set of chromosomes. And in order to do that, your DNA has to be copied. In order for your DNA to be copied, the chromosomes have to unzip and form new chromosomes. So the DNA has to be able to unzip itself or to pull apart to make a new strand. And in order to do that, the bond between the two strands has to be weak. So DNA is actually copied in a process called DNA replication. It takes place during the synthesis phase of the cell cycle, or the S phase. And remember, this has to take place because we're making two new cells. Each cell needs its own set of chromosomes. This replication is carefully controlled by enzymes. And enzymes are job doers. They do a lot of things in your bodies. They help break down starch, they help break down fat, and in this case, they're going to help DNA make a copy of itself. In this case, what's actually happening, the enzyme unzips the DNA and synthesizes new strands to pair with the original strand. So if you look at the diagram, we had some DNA, A to T, C to G. An enzyme is going to come along and unzip that, so you can literally see the AAC on one side and the TTG on the other. They're pulled apart. And then in red is the new DNA. So the A to T, C to G, A to T, C to G. And the circle there, that's that centromere, which holds the two. Now we have a chromatid. And eventually they'll be pulled apart in mitosis, and you have the two new cells with the proper amount of chromosomes. And black was the original strand, and red is the new strand synthesized by the DNA. Remember, the enzyme is what actually helps unzip it. So those weak hydrogen bonds are important so that this process can take place. To decode the genetic information in DNA, in other words, to read the genes and get the genes to do what they need to do, the cell uses a molecule called RNA, ribonucleic acid. In ribonucleic acid, the sugar is ribose. RNA is a molecule that carries out the instructions that are coded in DNA. So DNA has this code, A, T, G, G, C, G, A, whatever it may be, and then gives this code to the RNA, particularly the messenger RNA or mRNA, and it's called that because it is what reads the message. I tell students that DNA is like a dumb spy. He has this code. He's supposed to be able to read it, but he can't. He's a dumb spy. He cannot read his code. So he has to give it to the smart spy, who's the messenger RNA or the mRNA. And this smart spy is able to read the message. The messenger RNA can read the message. And then from that, it's going to make amino acids, which then make proteins, which then are your genes and do all the jobs in your body. So messenger RNA is able to read this message, but it has to go to a safe house to do this. The safe house is going to be the ribosomes. And if you remember from earlier, the ribosomes make protein. And that's what these codes are that are in DNA. Your genes are codes for protein. So DNA has the code for the protein. He cannot read the protein, gives it to the mRNA. mRNA needs to read the code and do what it needs to do. To do that, he has to go to the ribosomes. He goes to the ribosomes, reads the code, makes the proteins. Proteins give you your eye color, your hair color, help you digest your food, and so forth. There are some differences between DNA and RNA. The sugar in RNA is deoxyribose. The sugar in RNA is ribose. DNA is double-stranded. RNA only has a single strand. DNA has thymine. RNA has uracil. So DNA is in the nucleus. mRNA is in the ribosome. So here we have some DNA, A to T, T to A, G to C. DNA cannot read the code, has to give the code to the messenger RNA. So I take TAC, and the T is going to make an A, and you know that happens. The A would normally make a T, but RNA cannot make T's, so it made a U. The C makes a G. So we're giving this code, it's still the same code, we're just giving it to the messenger RNA, and instead of writing in T, we have to write in U for uracil, because mRNA cannot make thymine. It makes uracil instead. It does the same job as thymine. So now that the messenger RNA has the message, it will go to the ribosomes to decode it to make a protein. Transcription. The process that I just did was transcription. The process by which mRNA is made from DNA, or DNA is rewritten into mRNA. Transcription is carried out by RNA polymerase, an enzyme that connects to DNA and helps DNA make the RNA. It's helping the A turn into a U, the T to A, the C to a G. RNA polymerase helps produce a strand of RNA by matching the base pairs from DNA. If we have one strand of DNA, AACT, RNA polymerase would help make UUGA. There's your messenger RNA. Because remember, A's would normally go to T if we were making DNA, but to make RNA we have to go to U's, C's go to G, T's go to A. Messenger RNA now carries the copy of the coded instruction, or this gene. It will carry it to the ribosome to read it and to make the protein that is needed for your body to survive. Okay, so here's an example. And we will take this side here. Change the color of the pen. Okay, so we'll take this side here. T goes to A goes to U, G goes to C, G goes to C, T goes to A. There is our messenger RNA, AUCCA. It's only one-stranded. It will take off to the ribosomes. It will be read. It will make a protein. There are special stop and start sequences so that DNA knows exactly what chunk to read to make a gene. These sequences are determined exactly which part of the DNA will be copied into messenger RNA. Not all of your DNA codes for something. There are junk DNA. The junk DNA does nothing. It's only segments of your DNA that code for something. So your DNA has to know exactly when to start, and it will read it, and stop. Gives it to the messenger RNA. The messenger takes off to the ribosomes to make the proteins. So DNA codes for proteins. RNA actually makes the proteins.